the biomacromolecules, we had discussed that the polysaccharides, nucleic acids, proteins and lipids are the part of these biomacromolecules, but strictly when we consider lipid is not an biomacromolecule based on the molecular weight that is present. But now let us discuss about various types of biomacromolecules, starting with nucleic acids. So what are nucleic acids? The DNA and RNA. DNA, which is acting as a genetic material, and in few cases the RNA will also be acting as genetic material, but in general we say RNA is a non-genetic material. Those two are combinedly called as a nucleic acids. So before going to what are the two types of nucleic acids, now let us first discuss what is the composition that is present in nucleic acids. So nucleic acids are made up of the strands. We can call them as polynucleotide strands. These are made up of polynucleotide strand. And these polynucleotide strands, they contain nucleotide. So such many nucleotides are joined together with the help of bonds to form into a long and elongated structure called as a polynucleotide strand, or simply to say nucleic acids are the polymers of nucleotides. That is, many nucleotides are forming into a one single structure called as a nucleic acid. Then what is a nucleotide? Nucleotide is made up of nitrogen base, a pentose sugar and phosphate group. So one nucleotide is made up of one nitrogen base, one pentose sugar and one phosphate group. So the ratio between these three will be 1:1:1. Then what is a phosphate group? The PO4. Then pentose sugar, 5 carbon containing compound is called as a pentose sugar, and nitrogen bases, they are purines and pyrimidines. Let's start with pentose sugar. So here two types of pentose sugars are there. Those are deoxyribose sugar and other is ribose sugar. It is C5H10O4, whereas ribose sugar is C5H10O5. So that is one oxygen less in the deoxyribose, deoxy, that means one oxygen is less here. So if you see the molecular organization structure of this one, it's a ring structure with 5 carbons, and the 5th carbon is out of this ring structure. It forms CH2OH. So the valency of the carbon is 4. It is balanced here with 4 valencies, 4 bonds. Here also the valency of carbon is balanced, it is a 4. Here also the valency of the carbon have been balanced with 4. So here C5, 5 carbons are present and 4 oxygens. So 1, 2, 3 and 4 oxygens, and hydrogens 1, 2, 3, 4, 5, 6, 7, 8, 9 and the 10th one here. So total 10. So this type of molecular structure is there for the deoxyribose sugar. Whereas if you see the ribose sugar, it is structurally same to that of deoxyribose sugar, but it is differing in the position of the second carbon. So here this is the first carbon, second carbon, third, fourth and the 5th carbon. So here first, second, third, fourth and fifth. So first carbon is same with the same hydrogen and hydroxyl group on it, but here in the second carbon hydrogen and hydroxyl group are there, but here two hydrogens are present. So here the deoxyribose sugar and the ribose sugar are differing in the second carbon position. Where if you see the third one, hydrogen and hydroxyl same, and the fourth one hydrogen and here also hydrogen, fifth one CH2OH, CH2OH. The same composition is there, but they are differing only in the second carbon. So these are the two pentose sugars where 5 carbons are present, which are the cyclic molecules, with the fifth carbon is present out of the ring structure. In this one, the fifth carbon and the third carbon, so when during the formation of polynucleotide, they are binded with the phosphate molecules. So that structure will be seen when it is forming into a molecular organization. Then next are nitrogen bases. Here there are two types of nitrogen bases are there. Those are purines and pyrimidines. Actually, in the nucleic acids, there are 5 types of nitrogen bases are there, which are categorized into 2 groups. Purines or adenine, guanine, which is represented as capital A as adenine and capital G as guanine. Pyrimidines are cytosine, represented as capital C, thymine, which is represented as capital T, and uracil, which is represented as capital U. So here total nitrogen bases are 5 types in the nucleic acids. Which are being categorized into 2 groups, and purines are actually all these nitrogen bases are heterocyclic compounds. All these are heterocyclic compounds, and here purines contains 2 carbon nitrogen rings. 2 carbon nitrogen rings, whereas pyrimidines contain only 1 carbon nitrogen ring. Basically, we can show the ring structures like this. 2 different types of rings are there. Whereas here only 1 ring. We call dicyclic and monocyclic compounds. Dicyclic and the monocyclic compounds. Likewise, this nucleic acids contains the 2 groups of nitrogen bases. Then in the formation of the nucleic acids, the purines and pyrimidines will combine with the phosphate molecules and also with the sugar molecules. In that process, they form either the nucleoside or the nucleotide. So let's first discuss nucleoside. Nucleoside is the one which contains one nitrogen base plus pentose sugar. Here basically nitrogen base means consider adenine. Adenine plus pentose sugar. If it is a deoxyribose sugar, we call deoxyadenosine. This is the name of one nucleoside. In the same way, guanine plus deoxyribose sugar, it is called as deoxyguanosine. Deoxyguanosine. Cytosine plus deoxyribose sugar, it is called as deoxycytidine. Deoxycytidine. Then thymine plus deoxyribose sugar. It is called as deoxythymidine. Deoxythymidine. These are the 4 types of nucleosides which are present in DNA molecule. Next one, adenine plus ribose sugar. It is called as adenosine. So here there is a difference between adenosine and deoxyadenosine. Here in deoxyadenosine, deoxyribose sugar is present, whereas in adenosine the ribose sugar is present. And guanine plus ribose sugar, it is called as guanosine. Cytosine plus ribose sugar, cytidine. Here in RNA, thymine is absent. In place of thymine, it contains uracil. Uracil plus ribose sugar, uridine. These are the 4 nucleosides which are present in RNA molecule. Likewise, there are a total of 8 types of nucleosides are present in a nucleic acids. Then next are nucleotides. Nucleotide, as we said, that nitrogen base, one nitrogen base plus one pentose sugar and one phosphate group. Here if you see the combination of which type of pentose sugars are present, first one adenine plus deoxyribose sugar plus one phosphate. It forms deoxyadenylic acid. Deoxyadenylic acid. Then guanine plus deoxyribose sugar plus phosphate. Deoxyguanylic acid. Deoxyguanylic acid. Then cytosine plus deoxyribose sugar plus phosphate, deoxycytidylic acid. Deoxycytidylic acid. Then thymine plus deoxyribose sugar plus phosphate, deoxythymidylic acid. These are the 4 types of nucleotides which are present in DNA molecule. Then adenine plus ribose sugar plus phosphate. It is adenylic acid. The difference is only here the type of ribose sugar or the type of pentose sugar. Based on that one, they are differing here. Then guanine plus ribose sugar plus phosphate, that is guanylic acid. In the guanylic acid also, it is differing from deoxyguanylic acid based on the type of pentose sugar. Then cytosine plus ribose sugar plus phosphate, cytidylic acid. So this is also differing only in the nature of the pentose sugar. Then uracil plus ribose sugar plus phosphate, uridylic acid. These are the 4 types of nucleotides which are present in RNA molecule. So in total we can see 8 types of nucleotides in the nucleic acids. That is 4 types in DNA and 4 types in RNA molecule. In a nucleic acid, we say that a polymer of nucleotides are present, and these nucleotides are binded to the adjacent nucleotides are binded with each other with the help of phosphodiester bonds. That is, one ester bond is formed between the carbon of pentose sugar with the phosphate, and the same phosphate is binded to the next successive pentose sugar. So as a 2 ester bonds are formed, we call it as a phosphodiester bond. So likewise, the bonds are formed to produce a polymer of nucleotides. So as we said that nucleic acids are of 2 types, and these nucleic acids are DNA and RNA. Let's start with the first one, DNA. Deoxyribonucleic acid. This is acting as a genetic material in majority of the living organisms, both in the prokaryotic and also in the eukaryotic organisms. So the DNA will be generally circular in prokaryotes, but sometimes it may be linear also. In majority of the eukaryotes, it will be having a double helix model. So this model was proposed by Watson and Crick. Double helical model or double helix model. So here, with the help of various other scientists like Wilkins and Franklin, so they had awarded, Watson and Crick were also awarded the Nobel Prize for the double helix model proposed for DNA molecule. So let's see what's the organization that can be that is present in the double helix model of DNA. Likewise, we can see the 2 strands. So this is one strand of DNA and this is second strand of DNA. So in general, the DNA will be double stranded. Of course, exceptions like pi into 174, which contains single stranded DNA. So here in these 2 strands, one strand will be on one side we call it as a 5' end, and the opposite side is called as a 3' end, and in the second strand this side is called as 3' end and the opposite side is called as a 5' end. Why they are so called as 5' and 3' means, actually in the corners the pentose sugars are present. The fifth carbon of the pentose sugar is free here. The 5th carbon of the pentose sugar is free, so we call it as a 5' end. And if the 3rd carbon of the pentose sugar is free, that is on the opposite side, then we call this as a 3' end. Simply to say here, this is a place where the 5th carbon of the pentose sugar is free, so we call it as 5' end, and in the same strand the 3rd carbon of the pentose sugar is free, so we call it as 3' end. And if we take the second strand here the 3rd carbon of the pentose sugar is free, so we call it as 3' end. And for the same strand, if you are moving like this, on opposite side the 5th carbon of the pentose sugar is free here, so we call it as 5' end. And the 2 strands are parallel to each other. Wherever we see, they are parallel and they are coiling around each other, and one is moving showing 5 to 3, the other is showing 3 to 5. So but actually both of them are showing 5 to 3. So because 5 to 3 moving in this direction, the other one is also moving from 5 to 3 in this direction. So both of them moving in the opposite, so we call it as a anti-parallel. The 2 strands are anti-parallel to each other. And what is the length of an DNA molecule? It depends upon the number of nucleotides which are added to the DNA molecule. So it is not a certain, the length is not certain, but we can estimate the length by taking the length of one coil. From this point to this point, it is a 1 coil length. It is of 34 angstroms or 3.4 nanometers in its length. So the length of one coil in a DNA molecule is 34 angstroms or 3.4 nanometers. Then in this part only, so it forms a one circle. So the angle of one coil is 360 degrees. The angle from this point to this point, if you cut and it forms a complete circle, it is showing an angle of 360 degrees. And in this one, so what is the radius, or what is the radius from center to the here, or the total diameter? It is of 20 angstroms or 2.0 nanometers of diameter. From this point, at any point, if we take from this point to this point, from this point to this point, so actually the distance appears to be very less here, but it's not like, because as it is coiling from one direction, we say that both of them are overlapping, but it is not overlapping. At every point they will be at an equal distance, that is parallel to each other. They never fuse at any point. So this type of coiling is also there, where not only the two strands will coil around each other, and they also coil around the central axis. This point is a central axis. This is a central axis through which on which the DNA will be showing its coiling nature. In one coil, on one side, this whatever the strand-like structure which we had drawn, that is the backbone of the DNA molecule, which is formed by pentose sugar and phosphate group. These two will combinedly form into a backbone of DNA. So like phosphate group, pentose sugar, phosphate group, pentose sugar, phosphate group, pentose sugar. Likewise, they form a backbone for this one. Phosphate, pentose sugar, phosphate and pentose sugar. On this side also, phosphate, pentose sugar, phosphate, pentose sugar, phosphate, pentose sugar, phosphate, pentose sugar, phosphate and pentose sugar. Likewise, it forms the backbone of the DNA molecule. And pentose sugar is binded to the, and one more thing I said that every sugar molecule is binded to two phosphates. So here this is one ester bond between the pentose sugar and the phosphate. This is second ester bond between pentose sugar and the phosphate. Likewise, the two phosphate bonds are present, so two ester bonds are present. So we call it as a phosphodiester bond. And the pentose sugar is binded to the nitrogen base. So consider it is adenine. Here on the opposite side of the opposite strand of DNA, thymine is present, and the bond present between the nitrogen base and the pentose sugar is called as glycosidic bond. Glycosidic bond is present between the pentose sugar and nitrogen base. The same here also, between pentose sugar and the nitrogen base, this is a glycosidic bond. And let's take, if here it is guanine, on this cytosine will be present. So here if it is thymine, here adenine. So likewise the bonding can be seen. So likewise we can see the bonding, and from this end to, from this point to this point, a total of these pairs will be formed. So total of 10 base pairs are present. So one from here, one from, so this forms a one pair, second pair, third pair, fourth pair, fifth pair. Likewise 6th, 7th, 8th, 9 and the 10th pair. In this, let's take adenine is there, pairing with thymine. If thymine is present, pairing with adenine, and if guanine is present, pairing with cytosine, and the cytosine pairing with guanine. Likewise the pairing will present. And here there is a scientist called as Chargaff. He had proposed that whatever the number of purines present on one strand of DNA, the equal number of pyrimidines are present on the second strand of the same DNA molecule, and vice versa. That shows that the purines and pyrimidines ratio in a DNA molecule will always be 1:1, and the pairing is present, so that pairing is called as complementary nature. So here always the pairing is present between one of the purine present on one strand of DNA and one of the pyrimidines present on the second strand, or vice versa. Here always if you take the purine and pyrimidine, one of the purine always adenine pairs with thymine pyrimidine, and guanine pairs with cytosine pyrimidine, and the number of hydrogen bonds which are present between A and T are 2 hydrogen bonds, between G and C are 3 hydrogen bonds. This type of pairing is called as a complementary base pairing, and always the number of adenines and the thymines will be equal, that is 1:1. The ratio between G and C is also always 1:1. Otherwise, A plus G by T plus C is equals to 1. This type of relationship was described by Erwin Chargaff, scientist, so we call it as a Chargaff rule. Then here if you take this example here, two hydrogen bonds, here also 2 hydrogen bonds, 3 hydrogen bonds, 3 hydrogen bonds, 2 hydrogen bonds. Here also, here we had drawn 5, and here also 5 base. This is one base pair or one nucleotide pair, second base pair or nucleotide pair, third base pair or nucleotide pair, fourth, fifth, sixth, 7, 8, 9 and 10. So in one coil we get 10 base pairs or 10 nucleotide pairs, or as it is 10 pairs we are saying, so it comes to 20 number. Otherwise we can write it as 20 nucleotides or 20 nitrogen bases. 20 nitrogen bases. It is not compulsory that one coil should contain this many number of adenines and this many number of thymines, or this many number of guanines and this many number of cytosines. It may be that the entire one coil may contain only adenine and thymine pairs, or entire one coil may contain G and C pairs, or sometimes both A and T pairs and G and C pairs are present. So here what is the minimum number of hydrogen bonds in one coil? So it will be 20 hydrogen bonds. How it will be a 20 means, I said the 10 base pairs are present. If all the 10 base pairs are A and T, that means 10 are present. 10 into between one pair 2 hydrogen bonds are present. So it comes to 20 hydrogen bonds. And if 9 A and T pair are present and 10th one will be G and C pair, the number of hydrogen bonds will become 21. And if the number of A and T pairs decreases, and if the number of the equal number of G and C pairs will increases. At the same time, the number of hydrogen bonds are also increasing. And likewise, let's take a small chart here so that it is easy to understand the number of hydrogen bonds present in one coil of DNA molecule. Consider in one coil AT pairs, GC pairs, number of hydrogen bonds. consider that what is the maximum number, as we say one coil contains 10 base pairs. If all the 10 are A and T, so that none others will be there, GC pairs will be 0. So 10 into each pair contains 2 hydrogen bonds. So 10 into 2, 20 hydrogen bonds. And if one number is decreased, so the decreased number is occupied by G and C, one pair of G and C here. So 9 into 2 pairs. So 18 plus 1 pair, 1 into 3, because one GC pair contains 3 hydrogen bonds. So here 18 plus 3, it comes to 21. The same way, if A and T pairs are 8, so the remaining 2 are occupied by G and C. So 8 into 2, 16, 2 into 3 hydrogen bonds between each base pair. So 2 into 3, 6, 6 plus 16, it comes to 22. So as the number of A and T is decreasing, equal proportionately the G and C pair number will increase, and the hydrogen bonds number will also increases. So if it is 7, here it is 3, 7 2 ja 14 plus 3 3 ja 9, that comes to 23 hydrogen bonds. Here it is 6, the remaining 4 will be of G and C. So 6 into 2, 12, 4 into 3, 12. So it comes to total of 24 hydrogen bonds. Then 5, equal number, 5 2 ja 10, 5 3 ja 15. So it comes to 25. Here, if equal number of G and C pairs are present in one coil of DNA, what is the number of hydrogen bonds will be? It is middle, average number will be, that is 25. Then if it is 4, G and C pairs will be 6. 4 2 ja 8 and 6 3 ja 18. It comes to 26. Then here the number is 3. A and T pairs number is 3 and G and C pairs numbers will be 7. So 3 2 ja 6 and 7 3 ja 21. It comes to 27. Likewise 2 and 8. It comes to 28 hydrogen bonds. 1 and 9. It comes to 29 hydrogen bonds. Likewise, if all the 10 base pairs are G and C, then 10 into 3, 30 hydrogen bonds. So this is a maximum number of hydrogen bonds that can be present in one coil of DNA molecule. The minimum number is 20, the middle number is 25, and the highest number will be 30.